Welcome to Kalak Igniting the Young Minds. Let's begin the chapter. The cell cycle, cell divisions. Are you aware of all the organisms have even the largest and the starts their life form from a single cells? You have wondered how the single cells goes on from such large organisms. Growth and reproduction are characteristics of cells. Indeed, living organisms. All cells reproduce by dividing into two, with each parental cells giving rise to two daughter cells. Each time they divide, their newly formed daughter cells can themselves grow and divide and give rise to a new population that is formed by the growth and divisions of a single parental cells and its progeny. In other words, such cycles of growth and divisions allow a single cells to form a structure consisting of millions of cells. Cell cycle. The cell division is a very important process in all living organisms. During a cell division, DNA replication, cell growth takes place, and all this process, the cell division, DNA replication, cell growth, hence have taken place in a coordinated way to ensure a correct division. Formation of progeny. Cells contains intact genome, the sequence of events by which a cell duplicates its genome, synthesize the other constituents of the cells, eventually divides into two daughter cells is called termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events during a cell divisions. These events are themselves under genetic control. The phases of cell cycle. A eukaryotic cell it is illustrated by the human cell in a culture. These cells divide once in approximately 24 hours. However, duration of cell cycle can vary from organisms to organisms, also from cell type to cell type. Yeast, for example, can progress through a cell cycle in about 90 minutes. The cell cycle is divided into two classes, two basic phases, interphases and M phase, that is mitosis phase. The M phase represents the phase when the actual cell division of mitosis occur, and interphase represents the phase between the two successive mitosis phase. It's a significant note that the 24 hours of average duration of cell cycle of a human cell division proper lasts only for about an hour. The interphase lasts only for about 95% duration of cell cycle. The M phase starts with a nuclear division, corresponds to a separation of daughter chromosome. Karyokinesis usually ends with the division of cytoplasm, cytokinesis. The interphase, called as resting phases, is the time during which a cell is preparing for the divisions by undergoing both cell growth and the DNA replications in orderly manner. The interphase is divided into three further phases. G1 phases, gap 1, S phases is a synthesis, G2 phases is gap 2. The G1 phases corresponds to interval between mitosis and initiation of DNA replications. During G1 phases, the cell is metabolically active and continuously growth occurs does not replicate its DNA. The S or synthesis phase marks a period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cells doubles. If the initial amount of DNA is noted to be 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell has a diploid or 2-year number and the chromosome is G1, even after S phase, the number of chromosomes remains same, that is 2N. In animal cell, during S phase, the DNA replication begins in the nucleus. The centriole duplicates in cytoplasm during G2 phases, and proteins are synthesized in preparation of mitosis while cell growth continues. Stem cells in the adult animals do not appear to exhibit divisions, that is the heart cells. Many other cells divide only occasionally, has been needed to replace the cells and have been lost because of injuries or the cell death. These cells do not divide further exist as G1 phases to enter in the inactive stage called as quiescent stage, that is Z0 in the cell cycle. Cell in this stage remains metabolically active but no longer proliferating unless on on to depend on the requirement of the organism. In animal, mitotic division, cell division is only seen in diploid somatic cells. However, there is a few exceptions to which the haploid cell divisions by mitosis. However, the male honeybee against us, the parents can show a mitotic division in both haploid and diploid cells. From your recollection of the examples of alternation of generations in the plants, identify this plant species and stages at which mitosis is seen in the haploid cells, the young phase. It is the most dramatic period, and the cell divisions involves a major recognition of virtually all component of the cell. Since the numbers of the chromosome in the parents and the progeny cells are same, it is called as a equational divisions. Though the conventional mitosis has been divided into four stages of nuclear division, karyokinesis, it's very essential to understand cell divisions, is a progressive process. A very clear cut line cannot be drawn between the various stages. Karyokinesis involves the following four stages: prophase, metaphase, anaphase, telophase. Prophase is the first stage of karyokinesis of mitosis, following the S and G2 phases of interphases. The S and G2 phases and the next DNA molecule formed are not distinct but are intertwined. Prophase is marked by initiation of the condensations of chromosome material. The chromosome material become untangled during the process of the chromatin condensation. The centromere, which had been undergone duplications during the S phase interphase, now begin to move towards opposite poles of the cells. The completion of prophase can thus be marked by the following characteristic events. The chromosome material condensed to form a compact mitotic chromosomes. Chromosomes seen only is composed of two of chromatids attached together at the centromere. The chromosomes undergo duplication during interphase, begins to move towards opposite poles, and each centromere radiates out a microtubules called asters. Two asters together with a spindle fiber forms a mitotic apparatus. Cells at the end of the prophase, when viewed under microscope, do not show a Golgi complex, endoplasmic reticulum, or the nucleus than the nuclear envelope. Metaphases. The complete disintegration of the nucleus envelopes marks the start of a second phase of the mitosis. Hence, the chromosome spreads to the cytoplasm of the cell. By this stage, the condensation of chromosome is completed and they can be observed clearly under the microscope. This then is a stage at which a morphology of chromosome is easily studied. At this stage, the metaphase chromosome is made up of two sister chromatids which are held together by the centromere. The small disc-shaped structure at the surface of centromere are called as kinetochores. These structures serve as the sites of attachment of spindle fiber at the chromosomes are moved into positions at the centers of the cells. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid, each of the chromosomes connected by the kinetochore to the spindle fiber from one pole, and its sister chromatids connected by its kinetochore to the spindle fiber form the opposite poles. The plane of alignment of chromosome at the metaphase is referred as metaphase planes. The key features of metaphase are spindle fibers attached to kinetochore of the chromosome. Chromosome move towards the spindle equator, get aligned along the metaphase plates along the spindle fiber of both the poles. Anaphase. At the onset of anaphase, chromosome arranged at the metaphase plate it splits simultaneously to daughter chromatids, now referred as daughter chromosomes of the future daughter nuclei, begin the migration towards the two opposite poles. At each chromosome moves the equator plates and the centromere of each chromosome remain directed towards the poles. Hence the leading edge and the arms of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key steps. The centromere splits, chromatid separates, chromatids moves to opposite poles. Telophase. At the beginning of the final stages of karyokinesis, telophase, the chromosomes have been reached their respective pole, decondensed and lose their individuality. The individual chromosome can no longer be seen, and each set of chromatin materials tend to collect at each of the two poles. This stage shows a following key events: chromosome cluster at opposite spindle poles, and their identity is lost in a discrete elements. The nuclear envelope developed during the chromosome cluster at each pole, forming two daughter nuclei. The nucleolus Golgi complex and ER forms. Cytokinesis mitosis accompanies not only the segregation of duplicated chromosome into daughter nuclei karyokinesis, but cells itself is divided into two daughter cells by the separation of cytoplasm and called as cytokinesis, at the end at which the cell division is completed. In the animal cell, achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cytoplasm into two. The plant cells, however, enclose relatively inextensible cells walls, therefore undergo cytokinesis by different mechanisms. In plant cells, a wall formation starts at the center of the cells and grow outward to meet the existing lateral walls. Formation of a new cell wall begins with the formation of a simple precursor called as cell plates that presents a middle lamellae between the wall of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria plastid get distributed between the two daughter cells. In some organisms, karyokinesis is also followed by cytokinesis. As a result of multinuclear condition arises to the formation of synthetic liquid endosperms of the coconut. The significance of mitosis. Mitosis, or the equational division, is usually restricted to diploid cells. However, in some of the lower plants and some social insects, haploids also divide by mitosis. It's very essential to understand that the significance of this division in the life of an organism. Are you aware of some of examples where you have studied about the haploid, diploid insects? Mitosis usually result in production of diploid daughter cells, identical geographical components, and the growth of multicellular organisms due to mitosis. Cell growth result in disturbing the ratio between the nucleus. The cytoplasm, therefore, become essential for the cells to divide and restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is a cell repair. The cells of upper layers of the epidermis, cells of the lining of the guts, the blood cells are being constantly replaced. The mitotic divisions and the meristematic tissue and apical and the lateral cambium result in continuous growth of the plants throughout the life. Meiosis. The production of offspring by sexual reproduction includes a fusion of gametes, each of the components haploid cell chromosome, gametes formed by a special diploid cells. The specialized kind of cells reduce the chromosome number by the half, results in the production of haploid daughter cells. This kind of division is a meiosis. Meiosis ensures production of haploid phase in the life cycle of sexually reproducing organism, thereby fertilization restore the diploid phases. We come across meiosis during gametogenesis in plants and animals. This leads to formation of haploid gametes. The key features of meiosis are as follows. It involves sequential cycles, nuclear and cell divisions called as meiosis 1 and meiosis 2, but the only single cycle of DNA replication. Meiosis 1 is initiated after parental chromosomes have replicated to produce identical sister chromatid at the S phase. Meiosis involves pairing of homologous chromosomes and recombination of two non-sister chromatids of homologous chromosome. Four haploid cells are formed at the end of meiosis 2. The prophase 1, meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1. In the meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2. Meiosis 1. The prophase 1, now the first meiotic division is typically longer, more complex compared to mitosis. It's further been divided into the following 5 phases of the chromosome behavior: leptotin, zygotin, packetin, diplotin, diakinesis. During leptotin, the chromosomes become gradually visible under light microscope. The comparison of the chromosomes continues throughout the leptotin, which is followed by second stage of prophase 1 of zygotin. During this stage, chromosomes start pairing together, and this process is association called as synapses. Each such paired chromosomes are called as homologous chromosome. Micrograph at this stage indicate the chromosome synapses is accompanied by the formation of complex structure called as synaptimal complexes. The process, a complex formed by the pair of synapses homologous chromosome, is called as bivalent or tetrad. However, they are more clearly visible at the next stage. The first two stages of the phase 1, relatively short-lived compared to the next stage, that is a packaging. During the stage, the four chromatid at each bivalent chromosome become distinct and clearly appear as a tetrad. This stage is characterized by the appearance of a recombination of nodules, the site at which a crossing over occurs between non-sister chromatids of a homologous chromosomes. Crossing over is the exchange of a genetic material between two homologous chromosomes. Crossing over is an enzyme-mediated process. Enzyme involved is called as a recombinase. The crossing over lead to recombinations of genetic material, two chromatids. Recombination between two homologous chromosomes is completed by the end of packaging, leaving the chromosome linked at the site of crossing over. The diplotin is recognized as a dissolution of a synoptimal complex. The tendency of the recombinant homologous chromosome of the bivalent separates with each other except at the sites of crossover. The X-structured X-shaped structure is called as chiasmata. The oocytes of the seven vertebrates and diplotin can last for months or year. The final stage of meiotic one phase is diakinesis, is marked by the terminalization of chiasmata. During this phase, chromosomes are fully condensed and meiotic spindles are assembled to prepare the homologous chromosome for separation. By the end of diakinesis, the nuclear disappears, the nuclear envelope breaks down. Diakinesis presents transmission of metaphase. Metaphase 1 is a bivalent chromosome aligned in the equatorial plates, and the microtubules from the opposite sides of spindle attach the kinetochore core of the homologous chromosome. Anaphase 1, the homologous chromosome separate while the sister chromatid remain associated with the centromere. The nuclear membranes and nucleolus appear. Cytokinesis follows, and this is called as dyad cells. Although there are many cases where chromosomes do undergo dispersion and do not reach the extreme extent state of the interphase nucleus, stage between two meiotic division is called as interkinesis and the generally short-lived. There is no replication of DNA during the interkinesis. Interkinase followed by prophase 2, such similar prophase than the prophase 1. Meiosis 2. Prophase 2 of meiosis 2 is initiated immediately after cytokinesis, followed by chromosome fully elongated. In contrast to meiosis 1, the meiosis 2 resembles normal mitosis. Nuclear membrane disappear end of prophase 2 and the chromosome again become compact. At this stage, chromosome align at the equator, microtubule of the opposite poles, spindle gets attached to the kinetochores of the sister chromatids, begins to simultaneously split the centromere of each chromosomes, holding sister chromatids together, having moved towards opposite poles of the cells, shortening of the microtubule attached to the kinetochores, and telophase 2, the meiosis ends at the telophase 2, by which the two group of chromosomes once again get close to by the nuclear involved. Cytokinesis follow the resulting in the formation of a tetrad and the four haploid daughter cells. Significance of meiosis. Meiosis is a mechanism by which a conversion of specific chromosome number of each species is achieved across a generation as sexually reproducing organism. Even though the process per the paradoxically results in reduction of chromosome number by half, it also increases genetic variability in the population of organism from one generation to next. The variations are very important in the process of evolutions. According to the cell theory, cell arises from pre-existing cell, the process by which the occurrence of a cell divisions after sexually reproducing organism starts its life cycle from a single cell zygote. The cell division does not stop with the formation of mature organism but continues toward its life cycle. The stages through which a cell passes from one division to that of the next one next is called a cell cycle. The cell cycle divides into two phases: interphases, a period of preparation of cell division, the mitosis phase, actual period of cell division. The interphases is further divided to G1, yes, and G2. The G1 period is a cell growth and carries out the normal metabolism. Most of the organelles duplicates along during this phase. Yes phase marks as the phase of DNA replication, chromosome duplications. The G2 phases is a period of cytoplasmic growth. The nitrogen is also mitosis also divided into four stages, namely prophase, metaphase, anaphase, telophase. Chromosome condensation occur during these phases. Simultaneously, centromere moves to the opposite poles and the nuclear envelope and the nucleolus disappear and the spindle fiber starts reappearing. The metaphase marked has alignment of chromosome at the equator plate. During the anaphase, the centromere divides, chromatids starts moving towards the opposite poles. One Once the chromatids of two opposite chromosomal elongation starts, nucleus and the nuclear membrane reappear. The stage is called as telophase. The nucleation divides and then followed by cytoplasmic division is called as cytokinesis. Mitosis thus is equational divisions, which the chromosome number of the parent is conserved in the daughter cells. In contrast to mitosis, meiosis occur in the diploid cells, which are destined to the from gametes, called as reductional division, since it reduces the chromosome number by half while the making the gametes sexual reproduction when the two gametes fuse and the chromosome number is restored into the value of in the parent. Meiosis is divided into two phases, meiosis 1, meiosis 2. In the first meiotic division, homologous chromosome pairs to form a bivalent and undergo crossing over. Meiosis 1 has a long prophase which is divided further to form five phases: the leptotene, zygotene, pachytene, diploidine, and diakinesis. During the phase of 1, bivalent arranges the unequated plate. It's followed by the anaphase 1, which is a homologous chromosomes move to opposite poles with both the chromatids. Each pole receives half of the chromosome number of the parent cells. The telophase 1 is a number the nuclear number and the nuclei reappear. Meiosis 2 is a similar to mitosis during the anaphase 2 and the sister chromatid separates that at the end of meiosis 4 haploid cells are formed. You are tuned into Kalang, igniting the young minds. Thank you.